Okay, so welcome again to the Complex Fluid Seminar Series. And we'll have Arezoo Ardekani in a minute. And coming up next week, we have Jonathan Rothstein and Hadi Mohammadgushki talking about micellar fluids. So I hope you can join us also next week. Okay, so today's speaker is Arezoo Ardekani from Purdue University, where she's a professor in mechanical engineering, newly promoted, I understand, so congratulations.
Thank you.
So Arezoo did her bachelor's at Sharif and then her master's and PhD at UC Irvine in mechanical and aerospace engineering. She then spent a couple of years as a postdoc at MIT with Gareth McKinley and took her first job at Notre Dame in 2011. She joined Purdue in 2014 and has been successfully promoted there. She's won a number of awards, NSF Career Award in 2012, the Presidential Early Career Award in 2016. So unlike anyone else I know, she's actually met Barack Obama, or at least shook his hand or something in the days when we could do that. And I guess that was Obama and not Trump.
Yes, it was Obama.
It's a lucky break. More recently, this year was the Metzner Awardee from the Society of Rheology. It's been elected fellow of the ASME and various other awards. So Arezoo is going to talk to us about motile microorganisms in complex fluids. So I'll hand it over to you, Arezoo.
Thank you. Thank you, Jan and Rob, for organizing this nice seminar series and for the nice introduction. So I'm going to talk, can you see my screen?
Yes.
Okay, great. So I'm going to talk about motile microorganisms in complex fluids. Co-authors of this work are Gaojin Li and Nikhil Desai, Wasim Sheikh, and Yuchen Zhang, who are former and current graduate students. And as we know, let me see if I can proceed. Okay. As we know, different microorganisms have different motility mechanisms as well as different cells. Sperm, for example, uses a very long flagella, and undulatory motion of the flagella leads to its propulsion. Some algal cells have two or multiple cilia, and metacoronal beating of these cilia leads to their movement. Some bacteria have helical flagella, single or multiple flagella, and their rotation will lead to their motion. And slightly larger organisms like protozoa have arrays of cilia, and coordinated motion of these cilia leads to their motion. One thing that is common between these organisms, like bacteria, sperm, eukaryotic cells, or even C. elegans, and some of the macro or nano robots that humans build to do experiments with, is their small size. Their size often changes from 1 micrometer all the way to 100 micrometer. And even if they're swimming in a fluid with viscosity very similar to water, the Reynolds number would be very small, and inertia would be negligible, and viscous effect would be dominant. As many of you have seen this video, this is the video by G.I. Taylor that basically is showing important properties of very viscous flow, time reversibility. If you have a diafluid in Quetzal and rotate the inner cylinder a few times, it looks like the dye blob has mixed, but if you rotate it backward, it's very much clear that it's just a stretch, and you can basically recover the original shape on just a small diffusion, basically smearing the boundaries of the dye blob. Also, this properties of viscous fluid is very important when it comes to the propulsion of microorganisms. And this was first discussed by the paper seminal work by Parcel, which basically mentioned that time doesn't matter and the pattern of motion is the same whether slow or fast. And this is clear if we think about a scallop, that in water it would swim due to inertial effect, and by opening and closing its shell at different rates, it propels forward. But the question is, what happens if we put it in very viscous fluid like corn syrup? Following Parcel's theorem, then this scallop cannot propel itself. And I didn't know actually Parcell did his bachelor at Purdue before I joined Purdue, but then of course later on he moved to Harvard and did PhD in different field, then electrical engineering, and then got the Nobel Prize for his work in NMR. But organisms know these properties of viscous flows and know that using flapping motion doesn't lead to any propulsion, even though in water they can move. In very viscous fluid, they would not be able to go anywhere. So they use some way of breaking symmetry. For example, some bacteria have helical flagella and left-handed or right-handed flagella where they break symmetry and are able to move in very viscous fluids. This is an example of E. coli, which has multiple of these helical flagella. And these flagella are connected to the cell body through a motor, which has some resemblance to mechanical motor. It has actually a stator and rotor, and once in a while, one of these flagella rotate in the other direction, they unbundle and they move in a new direction. So what I'm going to talk about today is swimming in complex fluids. And the reason is that this itself also has lots of applications. For example, some bacteria like H. pylori, which is a bacteria causing ulcer, moves in mucus layer covering the stomach. And by changing the rheology of the fluid, can manipulate and maneuver around and control its propulsion. Also, sperm move in mucus covering female reproductive tract, and that itself has a complex rheology. Even in ocean, there are particles called transparent exopolymer particles or oceanic gels, which are made of high concentration polymers and macromolecules, and high concentration of bacteria has been observed inside them. And they are basically nuclei for formation of biofilm on, for example, membrane desalination plant or other places, and they're referred to as protobiofilm. And important examples of bacteria moving or bacteria living in complex fluid is biofilm. When bacteria come close to the surface, they produce what is called extracellular polymeric matrix that protects themselves from a harsh environment. And when they go to when they are protected by this extracellular polymeric matrix and form biofilm, that's when their resistance to antibiotic increases dramatically. And most of infections are caused by these biofilm. They can form everywhere in catheters connected to the body to transfer body fluids, on artificial implants and medical devices, all the way to pipes and anywhere you can think of. If you think about the extracellular polymeric matrix, for example, this is from Pseudomonas aeruginosa biofilm. And if you look at its rheology, you can see that it has both shear thinning viscosity and also elastic effect is observed. So this is first normal stress difference, which is also following shear thinning behavior. So both elasticity and shear thinning can be important for these extracellular polymeric matrix. Relaxation time can be on the order of 1 to 100 seconds, and they can be made of a large molecular weights polymer. So this study of swimming in general is going back to the work of Taylor, which looks at propagating transverse waves of small amplitude on sheets. And the amplitude of oscillation here is A, and this is the undulation pattern that is imposed. And the way you can solve this problem and how Taylor solved this problem is looking at perturbation method when amplitude is very small, and this undulation and traveling wave basically leads to motion of these swimming sheet. And the swimming velocity then would depend on amplitude and angular velocity and the angular velocity of this wave and wave number. Then later in 1979, Chitari, and then later in 2007, Eric Loga extended Taylor's swimming sheet to look at the viscoelastic effects. And this relationship is obtained for small amplitude oscillatory sheet of infinitely long sheet when it's swimming in Olroyd B fluid. So this is also done using perturbation method, where you can then calculate the factor of both elasticity, here described by Deborah number, defined as relaxation times angular frequency, and also depends on the viscosity ratio of the solvent to solution viscosity. This study shows that viscoelasticity of the fluid hinders locomotion. So swimming velocity goes down in viscoelastic fluid. There are experiments, and this is a very old experiment, that they put bacteria in polymeric solution. This is shown in terms of fluidity, but if you think about this as inverse of viscosity, so in this direction, you're increasing viscosity and concentration of polymer, and swimming velocity goes up before it goes down. And so since then, there has been many studies looking at swimming in viscoelastic fluid to understand under what scenarios we have speed enhancement or speed reduction. So for example, if you have a finite swimming sheet of finite amplitude, then because of the stretching of polymer along this undulating sheet, enhancement has been observed. For three-dimensional swimming, it has been shown that because of the stretching of polymer near the head and tail, you still have this, you get actually swimming reduction, but is less than what you expect for example, two-dimensional Taylor swimming sheet. For helical flagella, also depending on the pitch angle and radius of the flagella, swimming velocity can go down or up. So what I'll start talking about is that okay, we know for viscoelastic fluid, basically depending on the geometry, we get swimming speed enhancement or reduction. But what happens if we have shear thinning properties of these fluids? So in order to look at that, I'll start by a swimming model, which is basically starting with Taylor swimming sheet of infinitely long flagellum of a small and large amplitude, and as well as the symmetric swimming and cases where amplitude of oscillation is increasing toward the tail or decreasing toward the tail. So these are referred to as kicker and borrower, where kicker would be more similar to sperm, where the amplitude of oscillation is increasing toward the tail, and borrower would be more similar to C. elegans, where the amplitude of oscillation is decreasing toward the tail. So in order to look at this problem, we look at two different constant equations. One is Giesecke's fluid, which we have both viscoelasticity and shear thinning present, where we have now momentum equation, and the stress itself is decomposed to solvent stress and polymer stress. The polymer stress is basically includes the stress relaxation, the nonlinear term due to, and alpha here is motility factor, which correspond to an anisotropy in the drag force acting on the polymer. When alpha goes to 0, we get overweight B fluid, and then we have polymer viscous stress. We have viscosity ratio being important, as well as the Deborah number. And if you think about now defining, for example, elasticity number, which doesn't depend on flow properties and only depend on fluid properties like relaxation time, viscosity, and density, and the geometry or size of the swimmer, you see, unlike the inertial effect, when size goes down and inertial effect is negligible, elastic effect won't be the same. So elastic effects can be even more important when you go to smaller size. So we use both Giesecke's fluid as well as Cora model or inelastic shear thinning fluid. And if you then basically choose N, the power law, and the alpha, you can get very similar shear thinning effect where one fluid would be inelastic and the other would be elastic. So we have the zero shear plateau, infinite shear plateau, and the power law decay in between. So for the case of Taylor swimming sheet, if this amplitude of oscillation is very small, then the solution can be obtained using perturbation method. But what is observed is that the swimming sheet doesn't change up to order of amplitude to the power 4. And that's what we also get here. But if you think about Cora fluid, then we see increase in speed, whereas that's not the case if you look at infinitely long flagella. For Roy B fluid, you have reduction in speed for both the small amplitude, which are shown with open symbols, as well as large amplitude oscillation, which are shown with closed symbols. But also for Giesecke's fluid, we see a reduction in speed, but Cora fluid has an increase in speed. So let's see what's happening. So let's focus on the results for Cora fluid. So everything that is shown here are inelastic shear thinning fluid, where we are looking at the velocity normalized by the one in homogeneous fluid and plotted as a function of Cora number, which itself is relaxation times effective shear rate, here being angular frequency times amplitude of oscillation. So when shear is small, we are in zero shear plateau, the behavior is like a Newtonian fluid. When the shear is very large and we are in infinite shear plateau, also most of these curves, the velocity decays to Newtonian behavior. Somewhere in between, we have a peak. There are some of these curves that you see is monotonically increasing, and all of them correspond to this circle symbol, which is corresponding to eta s being 0, or beta s, which was the normalized viscosity, or basically solvent viscosity, solution viscosity being 0. So that corresponds to the case when then you don't have these infinite shear plateaus, so they monotonically increase. But what is the case? Why do we see this speed enhancement in shear thinning fluid? So if you look at the normalized viscosity around the flagella, there is a region of larger shear close to flagella, which is reducing the viscosity. So basically, the flagellum is swimming in a corridor of a small viscosity fluid, and that leads to speed enhancement. So it's acting like an effective confinement. So we tested that by putting this flagella in a Newtonian fluid of lower viscosity that is now surrounded by higher viscosity medium. And we also get a speed enhancement in that case. So this speed enhancement increases as we decrease this viscosity ratio. So for viscosity ratio 0, you have infinitely more viscous outer fluid. So basically it's like swimming between two rigid walls. And for that case also, we have swimming enhancement, and that's the maximum swimming enhancement you can get. So in another biological parameter that is important for these swimmers, in addition to swimming speed itself, is the swimming efficiency and power expenditure. The power expenditure can be calculated knowing the stress and the velocity and integrating that over the swimmer. And here, what we show is that all the data we get from these different amplitude and different viscosity ratios can actually collapse into a series of curves that only depend on the power law. And we can explain that actually by looking at, by non-dimensionalizing this power itself in non-dimensional form would depend on the normalized viscosity, beta E here, and the normalized shear rate. And if we normalize now this power with the 1 in Newtonian fluid and divide it by 1 minus the viscosity ratio, then it follows the relationship we got from the curve fluid model for viscosity. And so if we have the effective shear rate, then we would know exactly how it scales. An effective shear rate turns out to scale with amplitude. So doing that, then we have exact relationship that scales all the data, and that's the reason all the data collapses into a series of curves only dependent on n. Later on, there was an experiment done by Arezo's group where they tested our scaling relationship. And so they did experiments with CL again. They put it in shear thinning fluid and measured with PIV and measured the power expenditure. And from that, they saw that the power is decaying exactly as we predicted earlier. So in all the examples I showed so far, the swimming was happening in a bounded domain. But in many of the examples I discussed, like sperms swimming in female reproductive tract or biofilm, bacteria are close to surfaces. So what is the role of boundary here? So here is a undulatory swimmer in Newtonian fluid that is moving forward. If you put it in Oroid B fluid, in a viscoelastic fluid, we get very large polymer stretching near the tail as well as near the head, which basically the swimmer kind of stalls. It will have a very large angle and effectively doesn't move forward. On the other hand, when we put it in shear thinning viscoelastic fluid or gaseous fluid, it, because of the presence of boundary, can take advantage of both worlds. The polymer stretching, it's bringing it close to the surface, it leads to traction toward the surface, and then shear thinning effect leads to region of large shear and a small viscosity around the cell, which then leads to swimming enhancement. So it goes fastest in shear thinning viscoelastic fluid, and that is basically because of the presence of the boundary. In many examples, these swimmers move in as in a suspension. We have large concentration of these swimmers interacting with each other, and they show collective motion. So there are these patches of large concentration cell and as well as large velocity fluctuations are observed. Here is contours of vorticity, for example. These patches, their size can be on the order of 10 to 100 size of the individual swimmer. And so most previous studies looked at this collective motion in Newtonian fluid. So we're interested to know how viscoelasticity of the fluid can affect these collective motion. So we looked at on these collective motion of swimmers, modeling these swimmers as rods. So now we are applying basically slip velocity on the surface of these rods to make them propel forward. And we can have pushers, which basically is bringing fluid from the side and expels it from front and back. And it's a better model for bacteria, and vice versa, we can have pullers. So for these pushers, we see these regions of large velocity fluctuations very similar to the experiments. And when we look at ORB fluid, for example, here, Deborah number of 2.5, that reduces these velocity fluctuations. For pullers, on the other hand, these velocity fluctuations are smaller for both Newtonian as well as viscoelastic fluid. And if you look at the two videos, you see something is very different, and that's the way they form cluster formation. So this is more clear in this slide, where we are showing pullers and pushers side by side. We see the cells are either parallel or anti-parallel for the case of pusher, but they have these heads-on contact and form this asterisk in the case of pullers. So we can quantify that looking at probability pair correlation function, where it shows the same thing. So if you look at zero angle and angle of pi between the cells, there are peaks there. Whereas in the case of puller, it's distributed because they are forming this asterisk at different angle. What we are showing here is the contours of polymer stress. So it tells us about polymer stretching that is local, very close to these swimmers along the length and toward the tail. So looking at the pair correlation function as a function of a radius, we see the peak is much larger for viscoelastic fluid for the case of pushers. So pushers will have increased clustering formation in viscoelastic fluid. On the other hand, if you look at the velocity correlation and how it decays, where it goes to zero, it tells us about size of these patches of high velocity fluctuation and high vorticity. And that is almost unaffected by the presence of fluid elasticity, though the velocity correlation itself is decaying more rapidly in the case of a pusher and viscoelastic fluid. For puller, is not much affected, both cases decay the same. And later there was an experiment in Suarez lab where they did sperm in viscoelastic fluid and compare it with sperm cluster formation in viscous fluid as well as the standard medium. And they saw that the cluster formation is increased in viscoelastic fluid, which is in agreement with our prediction for collective motion of these swimmers in viscoelastic medium. So now let us focus on single swimmer, pusher and puller. So pusher is bringing fluid from top and bottom or sides and actually expels it from front and back, and pullers does the opposite. And if we look at the velocity decay around these cells, most of them it decays as -1/1, but in viscoelastic fluid for the case of pusher, it's decaying more rapidly. And the way you can explain it is that the polymer stretching for the pusher occurs along the length of these pushers. So polymers are coming close to the pusher, they get stretched, but then later they leave the swimmer and then they go back to the equilibrium point, and then the streamlines expands and velocity decays. This is qualitatively similar to die-swell phenomena, where you basically have a fluid going through a tube, it gets a stretch inside the tube, and then when it expands, the polymers go back to equilibrium shape and it expands. For puller, though, we don't see this effect because now it brings fluid from front and back. So and polymer stretching is large toward the tail, so we do not see such an increased decay in velocity field behind the puller, unlike the pusher. And that is basically the reason we see pushers' collective motion are not much affected. Sorry, pullers' collective motion is not effectively changed as much, whereas pushers are. Having basically the entire velocity field and resolving the polymer stretching as well as velocity field, we can look at energy balance for the system. So we have kinetic energy that is balancing the elastic energy, as well as the power input by the swimmers into the domain. And that is balanced by the viscous dissipation and dissipation due to the polymer stretching. We can calculate all of these terms. And then we also define this local alignment parameter, which tells us how long these swimmers are. So let's just look at the energy fluctuation. So this is kinetic energy as a function of time for a Newtonian fluid for the case of a pusher. So we do see the time scale of fluctuations is on the order of the time scale the fluid elements are convected across the swimmer. So that's the swimmer length divided by velocity. And that is also correlated with the RC here is the size of these large velocity fluctuation patches, or basically where the velocity correlation goes to 0. On the other hand, is not correlated with local alignment local alignment parameter. So the cluster formation is a local effect, whereas those larger scale of velocity fluctuations are not. If we look at now viscoelastic fluid, we have kinetic energy and elastic energy that are anti-correlated, and that makes sense because we get the collective motion of these swimmers. They generate these larger scale velocity fluctuations. They stretch the polymer molecules. Part of that energy goes to elastic energy, so it reduces the velocity fluctuations, and then it reduces the elastic stretching of these polymers, and the cycle repeats itself. And the time scale of these fluctuations now in polymeric fluid is reduced, and here is close to the relaxation time of the polymer. Looking at the kinetic energy spectrum, we see this is kinetic energy as a function of wave number and dimensionless wave number, and we see a -8/3 scaling for the case of a pusher in a Newtonian fluid. And this is very similar to what experimentally was observed for suspension of Bacillus subtilis, as well as suspension of E. coli in experiments. So all of these show -8/3 scaling. You may say, okay, we saw this for small wave number, and these were observed for larger wave number. But if you look at the range, are the same. They're both from somewhere between 0.1 to 1. The reason is that experimentally you cannot resolve such a high wave numbers or a small wavelength that we can resolve computationally. And you may say, oh, this number looks different, but that's actually the same because they are plotting wavelength square times kinetic energy. So basically, if you just go back and plot kinetic energy, you would get the same -8/3 scaling. So now if you look at the pusher in viscoelastic fluid, the scaling is different. So basically kinetic energy is reduced at a small wave number or larger wavelengths because of the, and there's a energy basically transfer from larger scale to smaller scale. At larger scale, these polymers are stretched and reducing kinetic energy, and it's pumped back at smaller scale. If it was at Newtonian fluid, the viscous dissipation would have happened at every scale because it's a zero Reynolds number problem, or and basically dissipation happens at every scale. So far I talked about undulatory swimming sheet or swimming rods, but we also looked at other swimming model. Here, a model is called Swimmer, and it's a swimming model for volvox, which is basically moving by metacoronal beating of cilia. And this model was developed by Lighthill and Blake, and its propulsion can be modeled with surface deformation and slip velocity that you impose on the surface of this sphere. And the swimmer itself is the organism itself also has a spherical shape. So the slip velocity we impose is as follows, which is what proposed by Lighthill and Blake. And by changing the ratio of these two coefficients, you can get different swimming mechanisms. If the ratio is 0, basically you have only the first term, leads to propulsion of the swimmer, and it's more close to what non-rotating volvox generates, and the flow has a 4-aft symmetry. When the ratio is positive, you have a puller where it brings fluid from front and back and expels it from the side. This is in the lab frame of reference, and this one is in swimmer frame of reference when you get this recirculation zone. And pusher does exactly the opposite. The far field solution of these pullers would be close to algal cell, and far field solution of these pusher would be closer to some of the bacteria like E. coli. So in order to look at this problem, we started with a methodology, basically simplest way we can think of, which is looking at it analytically using perturbation method before getting to numerics, so we can learn something from the analytical results, and the solution can be obtained basically for a wider range of parameters. So here we have the effective shear rate, which is the slip velocity over the size of the swimmer, and the effective shear or characteristic shear is described by viscosity times that. We have Weisenberg number this time rather than Deborah number because now this is a steady surface motion. And we have viscosity ratio. And we use basically weakly viscoelastic fluid where we have the Newtonian contribution to stress, and then Weisenberg number is assumed to be much smaller than 1, and all the other terms are described in sigma. So we use regular perturbation, so a 0th order solution would correspond to swimming in Newtonian fluid, and 1st order solution tells us about contribution of fluid elasticity into swimming. We can use Lorentz reciprocal theorem that connects the solution of our problem of interest, in being a swimmer, and an auxiliary problem, which has exactly the same geometry, but can have a different boundary condition. So the force and torque acting on the auxiliary problem is related to the velocity and angular velocity of our problem of interest, which is a swimming swimmer. And then that is related to the stress boundary, the integral of stress from the auxiliary problem and its slip velocity on the surface. Because we have the slip velocity, we have the auxiliary problem solution, we can basically get the swimming velocity and angular velocity. So this is at 0th order, but at 1st order, we have a very similar relationship. But now on the right-hand side, we need to calculate this volume integral. But sigma is evaluated on 0th order solution, which only is a Newtonian solution, which we have. And then we need to evaluate also the gradient of the velocity from the auxiliary problem. So we started this looking at a 2D swimmer for which the auxiliary problem would be a cylinder next to a wall in a Newtonian fluid. And this problem was solved by Jeffrey and Onishi in 1981. You can also do this for a 3-dimensional swimmer, and the solution for 3D swimmer, you can obtain it. This is done, for 2D is done using bipolar coordinates. And for 3D one, there's a paper by Lee and Lille that can be used to get this solution, and that is done using biospherical coordinate. So now, because we have this solution analytically, we can look at the large parameter space. So this is the phase portrait for different distance from the wall and different orientation of the swimmer, where we can look at basically the trajectory of these swimmers. In the case of neutral swimmer, we get 2 fixed points, 2 saddle points in both Newtonian viscoelastic fluid, and they are not that different. But for puller and pusher, we get 6 fixed points. 4 of them are saddle points, but 2 are center trajectory, where the swimmer doesn't, when it goes away from the wall, doesn't come back because the angular velocity significantly reduces. Whereas in viscoelastic fluid, even though we have very small Weisenberg number, but still we see a dramatic difference because now the 2 center trajectory become unstable foci, where the swimmer undergo these spiral trajectories here, which can get closer to the surface. And we also get formation of these limit cycles. Whatever is in this region will eventually move through this orbit because of these limit cycle we see in elastic fluids. So the behavior is similar for pushers and pullers, but it's very different from Newtonian fluid. And a parameter that would be of interest for biologists would be the residence time, or the time these swimmers spend close to the surface, because that's when they form biofilms. So we quantify that using these phase diagrams, where we are looking at the position from the wall and the orientation of the swimmer. And these different colors show the ratio of residence time, the time they spend close to surface in a viscoelastic fluid normalized by the one in Newtonian fluid. And these red colors show the one that are spending dramatically more time in viscoelastic fluid compared to Newtonian fluid. The white color shows the one that don't come close to surface. The blue color correspond to the cases that the residence time is not much different, but every other color, basically the residence time increases. So if you look at the overall behavior of these swimmers near surfaces, the residence time increases in viscoelastic fluid, which is important in terms of biofilm formation. This analytical solution is valid for a small Weisenberg number. If you want to look at larger Weisenberg number, we can numerically look at this problem. So this is a solution for a neutral swimmer. We see polymer stretching. This is the contours of conformation tensor, and we see polymer stretching behind the swimmer. The trajectory is not quantitatively different, and the residence time is also not quantitatively different for different Weisenberg number for pullers, actually. But for neutral swimmer, we have peak in residence time, which happens around Weisenberg number 1. For pullers, the behavior is different in terms of polymer stretching because it's bringing fluid from front and back and expels it from the side. We see polymer stretching in the equator plane. But the swimmer that is most affected is pusher, which we have a stronger polymer stretching, and eventually actually it comes close to surface and it has a perpetual entrapment near the surface. So it cannot go away from the surface. As we increase Weisenberg number eventually for Weisenberg number of 1 and larger, it doesn't go away from the wall. And this separation distance with from the wall is dramatically decreasing with Weisenberg number. So if you look at the contribution of the force and torque, here I'm showing just the force from Newtonian fluid and polymeric fluid, we can explain that because from the contribution of Newtonian fluid, we have a positive force basically moving, that is because of the thrust generated by the swimmer, and it's moving the swimmer away from the wall, whereas the contribution of the polymeric fluid is in the opposite direction because of the stretching of the polymer, and that's attractive force towards the surface, so they come to balance, and eventually the swimmer moves at a distance from the surface. We can see the difference between the three scenarios in one slide. We are showing these little ellipsoids, which are their size of major and minor axes are proportional to eigenvalues of conformation tensor. And so the more elongated they are, the more stretching of polymer we have. So for neutral swimmer, we have stretching of polymer in the back of the swimmer. In the puller, it's bringing fluid from front and back and expels it from the side, so we get stretching in the equator plane. And for pusher, we have stretching of polymer everywhere, but it's much stronger toward the back of the swimmer. And so if it has a positive angle, then this stretching leads to a force towards the wall, and then thrust is generating force away from the wall, and they balance each other, and it gets trapped. So in all these cases that we discussed so far, we have basically looked at continuum solution, where we have solved either for Orwood B fluid, Giesecke's fluid, or core of constant equation. But for longer polymer chains, the size of these flagella can be as small as like 20 nanometer, and can be smaller than the radius of gyration of these polymers, and continuum solution would not hold anymore. So the question is, what happens in those cases? So in order to understand that, we look at this helical flagella, or E. coli is a good example, which has a cell body and a helical flagella, looking at how that affects the swimming velocity and dynamics of these swimmers. This is relevant to the experiments I showed much earlier, from different bacteria moving in polymeric solution, but also there's a nice recent work from Wilson Poon's group, where they looked at E. coli in polymers of different molecular weights. For smaller polymers, they see velocity decay, but for larger polymers, they see small enhancement in velocity. So the question is, what causes this speed enhancement for larger molecular weight polymers? So hypothesis is that the high shear near the flagella stretches the polymer molecules and results in lower microscopic viscosity that is experienced by these flagella compared to the cell body, and that leads to speed enhancement. So we started looking at this problem numerically to see if we can prove or disprove this hypothesis by visualizing the polymer stretching and calculating stresses and effective viscosity experienced by the flagella and cell body. So in order to solve this problem, we have this helical flagella that is rotating, and we model polymer molecules using spring dumbbells, using a Brownian dynamics. So the solution for the fluid is solved by solving the momentum equation. We have this source term, which is a force acting on the flagella through this Lagrangian grid, which we discretize for the flagella to get the flagellar rotation. And then we have the force coming from these spring dumbbells. The motion of these spring dumbbells is described by their hydrodynamic drag, elastic force, and then Brownian motion. And putting all of this together, so basically what we do is we get the flow field, we interpolate velocity on a Lagrangian grid, we get the velocity on each Lagrangian grid on the flagella, as well as at the location of each of these polymer spring dumbbells. We calculate the force coming from polymers as well as flagella. We add them as a source term to the momentum equation and recalculate the flow, and we move forward. So there are three dimensionless parameters that are now important in this problem. We have Deborah number, we have gradient number, which tells us about the length of the polymer over the length scale of flow. And we have the Peclet number telling us about convection over diffusion for the polymers. So first we validate the results against a continuum solution. So this is undulatory swimming sheet, infinitely long, basically Taylor swimming sheet that you saw the results earlier. For Oroid B fluid, we know the swimming velocity decreases. And for Brownian dynamics, we also see the same decrease in velocity, and the results agree, except for very large Deborah number, for which we expected the results to deviate from each other, because now spring dumbbells, if the Deborah number is very large, can experience non-uniform velocity gradient across its length, and the results can deviate from Oroid B fluid. So now looking at the helical flagella that is undergoing rotation. So this is the flagella cross-section, little circle that is moving on this circle if you look at the cross-section of it. And we're changing gradient number in this direction from very small number all the way to larger values. So we have polymer stretching behind the flagella, and the polymers are now polymer stretching is reduced, and the stress is reduced as we increase gradient number, and the polymers are more diffused. So let's see what's happening here. So here we are looking at velocity of this flagella and the torque acting on the flagella normalized by the one in Newtonian fluid. So one corresponds to Newtonian fluid. So for a small gradient number where continuum theory holds, like Oroid B or Fini P or Giesecke's fluid would hold, then you have a constant value for this normalized velocity or torque, which correspond to those continuum predictions. But at some gradient number, then we have decay in the values of those velocities and torques. And for the gradient number corresponding to bacteria flagella, which is here, we have basically values that are very close to Newtonian counterpart. So if we look at now viscosity experienced by the flagella, it's close to the solution viscosity for very small gradient number, but for very large gradient number, it decays and approaches solvent viscosity. So for large gradients, the polymer size of the polymer dumbbell is much larger than the flagella, and so they experience non-uniform shear, and they are stretched much less, and that's why they experience much less viscosity. So this is basically showing the two scenarios. For short polymer molecules see uniform flow field. You see the size of the polymer is much smaller than the scale over which velocity is changing. This is the velocity field generated by the flagella. For much larger polymer chain, the length scale of the polymer is on the same length scale as the length scale velocity changes, and that experiences a smaller velocity gradient, and consequently a smaller viscosity is experienced by the flagella. For cell body, though, that's not the case, because the gradient number for cell body would be larger, and so it's more close to actually solution viscosity rather than solvent viscosity that is experienced. So knowing that the cell body experiences solution viscosity and the flagella experiences solvent viscosity, we can calculate the swimming velocity of the flagella. And this is the prediction we get compared to the experiments. And in both cases, we get this very small swimming enhancement, and they agree well. So depending on how we are doing in time, I may talk about something, another topic that is related to the things that I talked about here. And I guess we have some time. So before I wrap up this section of the talk, I talked about swimming speed as well as behavior of these swimmers close to boundaries in shear thinning fluid, in viscoelastic fluid, and shear thinning viscoelastic fluid, and also looking at scenarios where continuum mechanics doesn't hold anymore. We need to look at the individual polymer molecules and Brownian dynamics. So now for the case of swimming in complex fluid, there are also other effects that has been studied in the literature that I haven't talked about. For example, polymers can affect the tumbling of the cells. Earlier in the talk, I talked about these flagella forming a bundle, and once in a while, one of the flagella rotate in the other direction and unbundle. So in that case, if polymers affect the tumbling, and they tumble less, then they could effectively move faster. It affects their translational or rotational diffusion as well. Bundling time can be affected by viscosity, and shear-induced normal stress can also affect the bubbling of bacteria and bundling time. So all of these effects are other roles that polymers can play on motility of these organisms. So to recap what I talked about so far, we talked about how shear thinning viscosity enhances swimming speed of undulatory swimmer. We talked about how fluid elasticity enhances wall attraction for different swimming model that we described. And we talked about collective motion of these cells and how fluid elasticity suppresses velocity fluctuation and increases the cluster formation and also reducing mixing in these suspensions. The topic that I wanted to talk to you about next is related to the motion of these microbes now close to interfaces, and talking about role of hydrodynamics and how it affects the microbe interface interactions. The reason we are interested in this problem was motivated by the Deepwater Horizon oil spill that happened 10 years ago. And basically millions of barrels of oil was released into the Gulf of Mexico. And microbes came to our help. So basically they were the first responder. Their concentration peaked after the oil spill, and they decomposed much of the oil. So if they were not there, we had even more environmental disaster after the oil spill. So the question is that whether hydrodynamics play a role in that bioremediation process. For example, we know for nanorobots near polystyrene spheres, they can get trapped. So do we get similar behavior if you have bacteria swimming around droplets? And also after the oil spill, 7 million liters of dispersants and surfactants were released into the Gulf of Mexico. The question is, how does surfactant play a role in these hydrodynamic interactions? So in order to study this problem, we look at oil droplets as spherical droplets that are much larger than the bacteria itself. We model the bacteria as ellipsoidal, having a spheroidal shape. And then we model these bacteria as a forced dipole. So this is the simplest model you can use for describing the flow field around the bacteria, which corresponds to the far field flow this bacteria will generate. And there are experiments that show that this far field solution or forced dipole is a good measure of basically, or good approximation to the flow field that is experimentally measured. So on the top is experimentally measured flow field around E. coli, and in the bottom is the solution to forced dipole. So also in this case, the Reynolds number is very small, so we have Stokes regime. So first, in terms of solution or the singularity solution to Stokes problems, if you think about force monopole or point force singularity, that's solution for far field solution for a settling sphere. And if we have that solution, then we would be able to get also higher order singularities like forced dipole. So a forced dipole is basically here a model for a swimmer because there's a drag acting on the cell body and a thrust generated by the flagella. And you can also include higher order singularities. For example, a source dipole will give us tell us about the effect of finite size. Forced quadrupole can tell us about asymmetric propulsion. And a ropelet dipole can tell us about the torque, equal and opposite torque that is generated by the rotation of the flagella and a rotation of the cell body, and the torque that is acting by the surrounding fluid. So if we have the solution to force point force singularity, we would be able to get all of these higher order singularities, and then also understand the behavior of each of these effects. The flow field generated by each of these point force singularities are very different, but then we can, because we are dealing with linear governing equation here, we can superimpose them and get the solution of interest. So if we had the solution for a point force singularity outside a surfactant covered droplet, the problem would have been easy, but that was not existent in the literature. And student Wasim Sheikh calculated the solution for 0.4 singularity outside the surfactant-laden droplet. So here the boundary condition you have is zero normal velocity on the surface. You have the continuity of tangential velocity. You have then jump in the bulk shear stress, which is balanced by the Margoni stress and interfacial shear stress. And the transport equation for the surfactant is described as follows, but here we make a number of assumptions, assuming the surfactant is insoluble and also non-diffusive, so we get rid of this term. So at the end, the transport equation will simplify to just a surface divergence of velocity on the surface being zero. So I'm not going to go through the detail of this derivation, it's given in this paper, but what I want to do is use that solution and then understand how microbes behave, how bacteria behave near these droplets. So once you have the image singularity that you need to put inside the droplet to satisfy the boundary condition, then you can get the swimming velocity and angular velocity of these bacteria by using Faxon law. So from Faxon law, the hydrodynamic induced velocity would be this velocity field from the image singularity and then higher order terms. And then for hydrodynamic induced rotational velocity, we have the curl of this velocity field generated by image singularity. And then the gamma here relates to the aspect ratio of these bacteria. The more elongated they are, the larger gamma is. And then it depends on the strain rate that this image singularity produces. And then we have higher order terms. So in having this, then we can solve for trajectory of bacteria. The position and orientation of these bacteria would depend on swimming velocity, this hydrodynamic induced velocity, as well as the hydrodynamic induced angular velocity. First, I'll show cases where we don't have any diffusive motion, but if you want to include diffusive motion, that can be added here in both translation or rotational diffusivity can be added here. So we have four dimensionless parameters that are relevant. We have dipole strength, we have the drop radius, we have the viscosity ratio of the inner fluid to outer fluid, and we have a viscosity ratio of the surface viscosity over exterior fluid viscosity. So this bacteria move around, and there is this hydrodynamic attraction coming from hydrodynamic induced swimming velocity or hydrodynamic induced velocity and angular velocity. But when it gets very close to the surface, then it doesn't penetrate the oil droplet, and we have this steric interaction that we apply as well. So now if we look at how the results look like, we see that a bacteria close to a small droplet come close to it, but it gets scattered. But for a larger droplet, it gets trapped. And this is hydrodynamic trapping. It's all due to hydrodynamics. We do not have any other effects here. So if you look at the phase space, you have position or distance from the droplet and the orientation of the swimming. For smaller droplets, there is no fixed point. So cells come close to the droplet and then escape. But there's a fixed point for larger droplets, and that's a saddle point. So all the cells that are initialized in this region, they get trapped, and all the cells that are initialized in this region, they escape. So basically, hydrodynamic trapping happens for a radius of droplet larger than this critical trapping radius. And this critical trapping radius depends on dipole strength. So that the larger that dipole strength is, the larger hydrodynamic effects we have. So this trapping radius goes down. And it scales as alpha square, where alpha is the dipole strength. And the viscosity ratio also matters. So bubble would have largest trapping radius. As you increase viscosity ratio going to oil all the way to rigid sphere, the trapping radius goes down. So you have more attraction to more viscous droplets. When we add surfactant, something interesting we see is that when the trapping radius for surfactant gets smaller, but even smaller than the rigid sphere. And that was initially puzzling to me, why we can get more attraction even stronger than a rigid sphere. So we looked at the velocity field generated by these surfactant-laden droplets in presence of these 0.4 singularities. And what we see is that when we have surfactant-laden droplet for a 0.4 singularity, the solution can be described in terms of surface solenoid velocity field, which correspond to clean droplet, and a surface irrotational field, which correspond to solid particle. So now when we look at the clean droplet, when we increase viscosity, both normal and tangential velocity goes up, and that leads to more trapping. But for the case of the surfactant covered droplet, if the point for singularity is normal to the surface, then this part of the solution goes away, and we have the solution to be identical to the solid particle. So what happens is for surfactant-laden droplet, then when you increase viscosity, the normal velocity goes up. The normal velocity stays the same, like a rigid sphere, but the tangential velocity goes up, and that leads to less trapping. So that's the reason the behavior is as we just saw. And so if now you add diffusive motion on top of that, you see trajectories of these bacteria around the droplet. Some of them get trapped, some of them escape in a clean droplet. If you add surfactant for exactly the same size droplet, you get much more of them to get trapped near the droplet. So in summary, basically, surfactant increases the trapping and hydrodynamic attraction to the droplets, and that's due to hydrodynamics. Trapping time can be described. This is the probability distribution function for trapping time. And this is for bubble. It has the lowest trapping time. For viscosity ratio higher, like oil, you get more trapping time. For the surfactant-laden oil droplet, it's even more. And surfactant-laden bubble has the longest, basically, trapping time. So we wanted to test this behavior also experimentally. So, but rather than testing it first with oil droplet, we tested with flat interface. For the case of a suspension of bacteria between two rigid walls, this experiment was done by Lauga groups and also others, where they saw hydrodynamic attraction toward the surfaces, toward the rigid surface. So this is number of cells at each distance from the wall, and you see increase in concentration near the top, bottom wall and top wall. And that's due to hydrodynamic effects. So now the question is, what happens for liquid-liquid interface and also when you have surfactants? So we tested this for Pseudomonas putida and also E. coli. So we we have the glasses slide in the bottom. We have these culture medium where bacteria are swimming in, and then on the top we have the interface. We tested with different interfaces, like from CO2 all the way to different oils on the top. And we tested CO2 instead of oxygen because some of these bacteria show oxytaxis, so we wanted to eliminate that. So what we see, we look at the concentration of bacteria at different distance from the wall, and we see hydrodynamic attraction and increase in concentration near the wall, and the concentration is much more near CO2, which is consistent with the theoretical explanation I was giving based on our solution for swimmers near droplets as well. If we look at the different fluid viscosities like dodecaine, mineral oil, or soybean, we see the PDF of bacteria distribution is following the same trend, is also predicted from our simulation, which is basically including hydrodynamic interaction and Brownian and a diffusive effect. And the reason we see all of these different viscosity ratio behaving the same is because we can also theoretically show that when viscosity ratio is large, then it doesn't matter. For CO2 though, it makes a difference because it has a very small viscosity ratio. The behavior also very well predicts the swimming velocity distribution for these cells, but also the radius of curvature. These, because we are using these higher order singularities, we're also including rootlet dipole, and we do see the circular motion near surfaces and interfaces, and that PDF of these radius of curvature also follows what we get from experiments. Theoretical exercise here is to use the same solution and see how now different bacteria will behave around, for example, liquid-liquid interface and gas-liquid interface. And what we see is that pullers get trapped near interfaces, even more than pushers. But something that is very interesting, and we haven't tested it experimentally yet, but it would be very interesting to see if that's the case, is that we see shorter bacteria go toward the gas-liquid interface, whereas longer bacteria would go toward liquid-liquid interface. So with that, I'm going to conclude. I'm showing this limerick because for a conference we attended a couple of years ago, we were asked to write this limerick, so I'm using that to summarize the second part of the talk. So thanks to hydrodynamic interaction, surfactant helped to increase bacteria fraction, consuming oil droplets, eating them like chocolates, as it's combined with chemotaxis reaction, though I didn't talk about the chemotaxis reaction in this talk. And at the end, I would like to talk all the people who have contributed. Most of what I presented was done by Gaojin Lee, who is currently postdoc at, he was a student in my group and currently postdoc at Cornell. Soon he will join Shanghai Jiaotang University. Nikhil Desai, who was a student and is currently postdoc at Ecole Polytechnique. Wasim Sheikh, who a graduate student who just graduated and will join UBC, and Yuchen Zhang, who is currently a graduate student. And I would like to thank all the sponsors who have supported financially these works. And thank you, and let me know if there's any question.
Thanks, Arezoo. Yeah, we've got a series of questions. The first one's going to come from Alexander Morozov, all the way from Edinburgh. Alexander, you there?
Right, thank you.
Yes, we can hear
Hi,
you.
Arezoo. Brilliant talk.
Hi.
Thank you very much.
Thank you.
I was wondering about the part where you talked about bacteria and polymer solutions, naturally. As you have mentioned our paper quite a number of times, I just wanted to mention that in Martinez et al., we've constructed a rather silly model of a bacterium swimming in two viscosities. One is being experienced by the flagellum, which is the essentially viscosity of water because of the effects that you were talking about, while the other around the body was the viscosity of the polymer solution because the body translation and rotation are just not strong enough to stretch polymers. However, this model predicts our experimental data brilliantly, bar one ingredient. We need to dial in the motor torque speed relationship. I was wondering whether you have that in your simulations or whether you can get it with just the constant torque.
So we also need to have the same relationship you mentioned. And what I want to clarify here is that we here for the flagella or cell body in a polymeric solution, we looked at them separately. So the part that I wanted to test was that whether flagella actually experiences viscosity close to solvent and cell body viscosity close to the solution or not. So basically we looked at them separately. So for those, we don't need any torque or any relationship. So basically we quantify things in terms of calculating, for example, the torque and velocity for different input velocity or angular velocity. But if we want to use the same resistive force model that you described, for that, then you would need to include that torque relationship that you just described. All right,
I see. Just a short comment. I'm on a warpath against the Schneider and Deutsch paper. As an introduction to the field, that was wonderful. But I mean, there is very strong evidence that increase is caused by small impurities in their polymer. So essentially bacteria eat those and speed up. And when you dialyze the solution, this effect goes away. Yeah, I agree with that. Though even in your paper and Wilson points, you also saw this increase in speed, though described by different physics, basically. Yes. All right, thank you.
Thanks, Alexander. Good to hear from you. The next speaker is going to be Ala. Let me just, Ala, are you there? Okay. Hi, Ala. You need to unmute.
Hello.
Hi.
Thank you very much for the presentation. Actually, if there is little slight yield stress of biological material or fluid, is there possibility for the bacteria, for example, like E. coli, to flow or to move or not? And another question, the head will experience stress different from the flagella. So what that affects as well? Thank you.
So the first part of the question, which was whether it would be able to move in yield of stress, that would depend on whether the stress that is generated by the flagellar rotation is larger than the yield of stress of the fluid. So I guess it depends on the yield of stress of the fluid that you're considering. It may or may not swim around. And the other question, I'm not sure if I got it correctly. So your question was whether in the case that they experience different viscosity for head and flagella, if that's the case for the yield of stress fluid as well. Is that the question you asked?
Yes.
Okay. In that case, if you have some microstructure, which then because of the different flow field basically generated by flagella and the cell body, then the microstructure would feel the uniform flow field generated by, let's say, flagella, or the non-uniform flow field generated by flagella, then yes, they would experience different effective rheology.
Okay, thank you very much.
There was a very nice paper, I think, last year by Duncan Hewitt and Neil Baumthorpe, looking at this sort of helical corkscrew going through a yield stress. You know, and basically the helix follows its own path, so it's very efficient to get through there. Just you know, if you're looking for something more on that, but I mean, I don't think they were really targeting bacterial motion, but it was just a very cool simulation that they had.
Yeah, thanks for mentioning.
Yeah, that's in JFM if people want to find it. Thanks a lot. Next question is going to come from Marco Polin, not Marco Polo as I was about to call him, but Marco, are you there?
Hey, that's almost.
Almost, yes.
Actually, yeah, in my lab they put Polo instead of Polin, but it doesn't matter. So thanks for the talk, it was very nice. I was wondering whether you have looked at the effect on metachronal waves of elastic interactions, like for example in epithelial tissues in the lungs. I don't know, I mean, it might be that you have seen it. And if I can also ask a second question on the Putida experiments, I was wondering whether you observe any difference in swimming speed between the two sides, the top and the bottom.
So regarding your first experiment, we haven't looked at that, but that's definitely an interesting problem of looking at the metacoronal beating of cilia and how that also affects transport of, I guess, particles as they get trapped in airways, for example. And there are some papers out there on those when they look at either two layers of different viscosity or different rheological properties, or one layer but having continuous change in rheological properties. But related to your second question, when you talked about Putida, we did get a slight difference also in swimming speed for the top and bottom boundary, where top was a liquid-liquid interface and then bottom one was wall-wall interface.
And would it be possible that then that contributes to some of the differences in concentration accumulation at the two sides?
They will be related. So both concentration and there's a slight change in velocity, both are due to hydrodynamics effect. So basically, from hydrodynamics point of view, in order to satisfy the boundary condition at wall as well as the surfactant-laden interface, you would need to include the image singularities. And then the velocity field those image singularities would generate at the location of the cell would be different. Depends on the, you can think of a viscosity ratio, or also depending on the boundary condition itself.
Great. Okay, thanks for your question, Marco. So the next question would have come from our favourite questionnaire, Cecile, but she's had to leave. And also, I think actually a couple of people in the chat have answered that question nicely as well. So if people are wondering where Cecile is, a question has been upvoted, but she had to leave. So the next question then will come from KVS Chaitanya. Apologies for mangling your surname. Thanks, Mark. I'm just going to... KVS,
yes, can you hear me now?
Yes, hear you perfectly, yes.
Thanks for the nice talk, Professor. It's like reading a nice review paper. So I have a question related to collective behavior. So did you compare your results related to collective behavior with the results obtained from continuum model simulations like active nematics? Because when you show the alignment of these pullers and pusher rods, I see some sort of a defects appearing even in your case. So did you try to compare this with the active nematics literature?
Yeah, you're quite right. You do see some of those defects as well. We haven't looked at the cluster formation or these parameters or defects from the active nematic literature, but looking at the studies of continuum approach for example, energy spectrum, like solving for the collective motion of bacteria and looking at energy spectrum, the scaling of the decay of energy, kinetic energy is very similar.
Okay, thanks. I have one more question. So the model that you have proposed for a forced dipole near a surfactant-laden drop. So did you look at the dynamics of a forced dipole confined in a fluid drop rather than outside?
For the forced dipole inside the droplet. So there is a recent paper that is like collaborative with a number of authors, and that's on the archive, and there, yes, that's described. And something that is already published on that is not forced dipole, but looking at the swimmer inside a droplet that we published a couple of years ago.
Okay, thanks.
So we've got the final question is going to come from Hamid. Hamid, are you there? You'll need to unmute. You there, Hamid? Are you there? Yes.
Hello.
Hi. Hello.
Hi. Thank you so much for the nice talk, Arezoo. I'm wondering about the coupling between the orientation and storage regime in your system. Could you comment on this? It's basically from the Weisenberg that you defined from your experiments.
So I just want to make sure I understood the question correctly. So you're talking about like the polymer stretching for different Weisenberg number and how that changes for the problems we described?
Yes, and also the transition between the orientation and chain alignment due to the flow, and also go to the stretch regime. This is something, is it the same way as the polymers, or is it the other thing that you want to make a comment? Yes.
So for the polymer stretching, so for example, what we see for the case where the flagella was in long polymer chain, we modeled as the Brownian dynamics. The polymer stretching is behind the rotating flagella, and we see that wake of large stretching of polymers behind it. But then as we increase the gradient number, or basically it's the size of the polymer chains, then we see that goes down, and the polymer stretching goes down, and then stress goes down. And that is one of the reasons that the viscosity that is experienced by those flagella is also reduced.
Yeah, but just one thing, your Weisenberg, it's about 1, yes? Correct?
So no, we varied Weisenberg number.
Oh, okay. Thank you so much.
Does that answer your question? Thanks, Hamid. So that's all the questions finished with. Ian, do you want to come in and say a few words before we finish?
No, thank you.
Okay. Ian, you there?
Yeah. Thanks, Arezoo. That was a wonderful talk, lots of detail, and I had a general question, really relating to your squirmers. So it's not really to do with anything you showed, but so I've seen people do mechanical experiments where you have helical tails and things like that. What about people doing mechanical experiments with squirmers? Does anyone do that, and how do they? How can you do that?
That's a very good question. So I haven't seen anyone building a mechanical squirmer. That would be quite challenging, I think. Though what I have seen is like people making like a couple of cilia, like adding motors or generating a couple of cilia moving around, but having whole array of cilia undergoing metachoral beating, I haven't seen such a work.
Right. Yeah.
But maybe, I don't know, I'm just thinking loud here, but maybe like, because we know we have these arrays of cilia on, for example, airways, maybe there can be kind of built out of those, and is still like having like some tissues that are still beating outside or like ex vivo, and then building something like that. I don't know. I'm just thinking loud.
Oh, I see. Stimulating them somehow.
Maybe electrically or somehow. Yeah.
Yeah. Okay. Yeah. 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 I mean, mathematically, some of this stuff, some of the things are similar to when you do peristaltic sort of pumping, aren't they? And they want, you know, I did wonder if there's some way to simulate that with, you know, mechanically with something peristaltic.
I guess for the cases, for not exactly the one that I showed, but for the cases that the propulsion is done by deformation, not the slip velocity, I guess you could, yeah, you could make it
right.
with static motion.
Yeah. Yeah. Okay. But the only other question I have is that photo's got a lot of people in it. What are the rest of them doing?
Oh, different things. Like a couple are looking at flow through purse media, and actually, this is the semester that I had the largest number of students. Like 4 of them already graduated, so now it's a little bit smaller.
Yeah, that was great. Thank you.
Yeah, superb. So it's a great talk, Arezoo. Thank you. So traditionally now, Ian and I give you a small round of applause, and then people give the congratulations through the chat. So thanks for a great talk.
Thank you.
And you've got to imagine that's applause from around the world, because you're having from England and also from Canada. So you know, so thanks everybody for joining. Hopefully we'll see you next week. I'm going to stop the recording.